like publicly, or we will also talk offline, I guess.
It looks like someone just commented one.
Okay. So Kirsten volunteered to monitor the chat. Let's see. I couldn't find the link to the textbook reference. Yes. Okay. So I will announce this once at 9:10. That was my mistake. And anyway, I'll explain it. And I'm still trying to figure out if I can make the textbook available to everyone or if people will have to buy it. I hope that we can make it available for free, but I'm back from the publisher.
I do think that it will be impossible. My personal thought is that it will be impossible for people to buy because Amazon is currently, like, I ordered something and they say I'll get it at the end of April.
Yeah. No. You would buy the ebook anyway.
Oh, got it. Okay.
Someone else just commented that there's an email saying the Cal Store is giving out free access to textbooks.
Okay, I don't know if this one is included. Anyway, I will do my best to see if I can get it made available for free. And the publisher made all of their teaching materials available, like the quizzes and stuff that I posted are a module that I downloaded from the publisher that they made available for free for this semester. And I thought that included the ebook. And I realized only when someone asked this morning that the ebook was not actually visible to you guys on the website. So on B courses. So I'm going to try to figure out how to make it available. Anyway, I'll wait till 9:10 to make this announcement just to make sure everyone's here because people are still joining in. Yeah, if we can't make it available online, yes, I could provide PDFs, but I think if the ebook is available, it's really quite nice. It has a lot of useful features, like you can highlight, you can mark it up like a regular book. I can even like go in and insert stuff if I want to add links or other comments and stuff. So you have a copy of the book. Valentina says she has a copy of it. Is that the 5th edition? This one? Okay, so that's great. So maybe if it is publicly available as a PDF, that's great. If we can get it as the Norton, as the ebook, like with their reader software, that would be even better because it has some really nice features for studying and integration with the B Courses site. So that would be the best option. So it looks like a few people have the book. That's great. Okay.
And we've had a couple people ask if the quizzes on B Courses are for grades.
So no, I mean, I didn't even mean to put those up there. And I saw some people started doing them even before the break, which was kind of remarkable. But so no, those are self quizzes. I think that there's a way that I can tailor those to like follow the material that I'm going to focus on more specifically. But that's one of the things I'm still trying to work out. So I have I'm going to be talking with the publisher's rep today, and hopefully we'll be a little bit more sophisticated about how to use this material. So, okay, it's now 9:10, so I'll just kind of repeat some of what I just said. So, as we've moved to this distance learning, I really thought it would be helpful for many of you if we followed the textbook a little bit more than I normally do. This is the textbook. I realize very few of you have a hard copy. I'm going to try. I thought I had made the ebook available through B Courses, but I only realized when someone asked this morning that that's not the case. There's a bunch of stuff on B Courses, but it's not the actual book. So I'm going to find out hopefully today if I can make the ebook publicly available. A couple of people in the class mentioned that they have copies, electronic copies of the book that they can share. And that would be sort of second best if we do that. But if I can actually make the ebook available through the publisher, it has nice features, like it allows you to highlight, and it allows more interactive teaching from the book. So some of you may have used this book before. It's an abridged version of Molecular Biology of the Cell, which is a really great book. It's got a lot of the same authors. And it's, I think, really nice for this class in particular, because this is sort of a very compacted introduction to cell biology, and this book is well tailored for that. Most of you have taken Bio 1A, hopefully, and have had some cell biology before. I'm not exactly sure how, it's been about a decade since I taught this course, and I'm updating what I've done in the past. If you feel like the level is not appropriate, you know, if you want me to be a little bit more high level, then we can, then please give me feedback and I will try to adjust as appropriate. It's hard to gauge in this format exactly what people know and so forth. And hopefully using some of these quizzes and other teaching aids provided with the book will allow me to assess where you guys are and adjust a little bit on the fly. So, all right, so I, you know, I'm a cell biologist. I graduated from Berkeley in, I won't tell you exactly when, but when I graduated, I had no idea what cell biology was, even though I'd taken a class called cell biology. And it's because it's really fuzzy. It's not like, you know, genetics is really the study of inheritance and how, you know, genetic information is transmitted from parents to progeny. Genomics is also sort of a better defined discipline, in that it is really the study of genomes, and the main tool of genomics is DNA sequencing. Cell biology is very broad and thus hard to kind of get a grasp on, because it really encompasses the whole field of cellular structure and function, which is pretty massive. And it's not, so it doesn't have it doesn't have a set of first principles, and it doesn't really have a core set of experimental approaches. I would say that one thing that really makes cell biology is the use of microscopy. And I'll talk a little bit today about why microscopy was such a defining, why the development of microscopy basically led to the entire kind of discipline of cell biology. Because obviously cells are small, and it really took microscopes to be able to start understanding what happens inside them. So basically anything to do with cellular structure, function, and self-assembly can be described as cell biology. And arguably cell biology started even in the 17th century, when the very first kind of microscopes were manufactured that enabled people to see individual cells. It was really sort of 19th century when people started looking at things inside cells and looking at their dynamics, and starting to describe things like mitosis and meiosis. And then in the 20th century, we saw a major revolution in microscopy, which is continuing today, that has led us to understand at a descriptive level many of the structures inside cells. And we are, you know, developing an increasing number of different tools to manipulate those processes and structures to understand at a molecular level how things inside the cell are built, how they work, how they interact with each other, how they take in information from outside cells and enable cells to communicate with each other, and so forth. So it's a very broad discipline, which makes it very hard to kind of encapsulate in a way, and no cell biology course can touch every aspect of cell biology. So we have to make, you know, decisions here about what are sort of the key topics to cover. And I've updated the syllabus. It's, you know, to reflect the fact that we skipped 2 lectures before the break because of the midterm. And also I put the lectures more in the order that they are, the topics in the order that they are in the book, just because I figure that way we won't have issues where I talk about something and realize that you kind of needed some foundation that we didn't get to beforehand. So let's see. There was a kind of mock kind of fight back in the days when I was a graduate student between geneticists and cell biologists, where geneticists think of themselves as being very precise, logical thinkers, and they describe cell biologists as fuzzbrains because there isn't the same kind of rigorous logic in cell biology. But in fact, cell biology can be extremely quantitative and rigorous. It's kind of a cultural joke more than anything else. And I'll share Yeah, okay, we'll stop there. So, as I said, like this is the book that we're gonna use, and I'll make as much of it available as possible. And I would encourage you to, read the chapters that I'm gonna refer to before lecture, just so that the lectures reinforce what you've already kind of read over. And let's see, anything else I want to say about that? And there are a bunch of kind of teaching tools within the book that allow you to sort of self-assess. And I'm going to try to implement those as we go, but I have to still learn how to do that. Okay, so the idea that all living organisms are constructed of cells really came from direct microscopic observation of plants and microorganisms, and this realization that no matter how large and complex organisms are on Earth, they are all built of these units that we call cells. And a cell is basically a self-replicating unit of life that is surrounded by a membrane, a plasma membrane, and I'll talk about membranes and their structure and function. But what's sort of magical about membranes is that despite being incredibly thin, they create a compartmentalized environment. They shut out, they prohibit the transport of many molecules, most large molecules and even most small molecules, and thereby can create a chemical environment inside a closed cell that can concentrate energy, can do magical chemistry, and can enable self-replication. So I just made a note at the bottom, since we're thinking about viruses a lot, that viruses are not really considered cells. They are not intrinsic, they are not autonomously self-replicating. They have to use the host machinery of a cellular life form in order to replicate. So in that sense, they are not living. Cells obviously have a huge diversity of shape and size and function. So these are just some examples in the textbook. And I think if it would be helpful for you guys to look at these scale bars. Most of the images in the book have scale bars because you see an image, and if you're a biologist, like, you might look at this and say, oh, that looks like a fission yeast, and you might mentally have a concept of how big it is. But these cells actually span a massive range of different size and morphology. So this is just an example. This is a neuron with its axon and all these dendrites. So it's a very specialized cell morphology, highly branched, and a specialized membrane. This is a ciliate. This is a genus of, or actually a whole clade of organisms that has really remarkable morphology. I'll show you a cool example in a few slides. This is the surface is a scanning electron micrograph of the petal of a snapdragon. This is a mammalian cell. This is crawling on a flat surface, and this is a fission yeast. And these are all different kinds of images taken with different kinds of microscopes, different kinds of ways of looking at, in most cases, just the kind of surface morphology of the cell. And then in this case, you're actually seeing into the cell using fluorescent tags. So we'll talk a little bit about different methods of visualizing cells, and hopefully you'll start thinking a little bit when you look at images of cells, like, what is it that I'm seeing? Because an image is always just a representation of a very complex thing, and sometimes you're looking at the surface, and sometimes you're looking at internal components. Okay, so this is just another kind of striking example of how different cells from the same organism, in this case, an ovum and sperm, these are cells that have sort of similar function, but extremely different morphologies. The ovum in most animals is the largest cell that the animal makes, and sperm are actually very long, but the head where the nucleus is one of the smallest nuclei that are made by animals. So just these are just examples to think about how cellular morphologies can be really variable to attain different functions. these are just kind of the same examples that I already showed you, but with labels. So I'll skip that over. But notice that the size scale. So micron is a millionth of a meter or a thousandth of a millimeter. And usually when we're talking about cells and the things inside them, we're talking about things on the micron to nanometer scale. So that, yeah. Okay. So I think you're all aware, and this has already kind of been relevant to other aspects of the course, like the genomics part in particular, but one of the core principles of modern biology is that all cellular life on Earth arose from a common origin. And so we talk about the tree of life as this branching tree because all organisms are related to each other. And the tree has sort of changed in its structure over the years. That the importance of the archaeal branch in particular has become much more evident over the last few decades. And this is just one representation of the tree. But in this course, we're going to be focusing really on eukaryotic cell biology. These are also cells, eubacteria, or bacteria as we call them, and archaea are actually much more numerous on Earth than the eukaryotes. And they actually have a much more diverse chemistry. That most of the chemical processes on Earth were had to have their origin in these prokaryotic forms of life. So chemically, they're much more versatile and diverse than the eukaryotes. But because we are eukaryotes, we're like out here on the little animal branch, we are particularly interested in eukaryotic cell biology. And also eukaryotic cells tend to be larger and more amenable to imaging. And so cell biology really got its start looking at eukaryotic cells. We're now increasingly aware through advances in microscopy that even these much smaller single-celled organisms often have a lot of internal structure that wasn't evident for many years. And there definitely is a thing of, studying archaeal and bacterial cell biology is definitely a thing. But for this course and for most of the field of cell biology, we're going to focus on the eukaryotes. Okay, so there are some key differences between these different branches of the tree. All prokaryotes are single-celled. They can form colonies where cells have specialized function, but the cells are independently kind of replicating. Whereas the eukaryotes are both single-celled organisms and multicellular organisms. And multicellularity, as I think Nicole has told you, is something that has arisen multiple times and independently in animals and plants and fungi. The prokaryotes and eukaryotes are also distinguished from each other by their replication. So all bacteria and archaea replicate through an asexual process. They can undergo conjugation, so they can sometimes exchange genetic information, but they don't do true sexual reproduction. Whereas almost all eukaryotes reproduce sexually, meaning you have two parents that contribute genetic material to their offspring. And we will cover that in a future lecture. There are a few eukaryotes that have lost the ability to reproduce sexually, and but they're kind of the exceptions. And I'll just put a couple of terms on here for reference. We talk about the last eukaryotic common ancestor as the organism that was shared as an ancestor by all eukaryotes, and that's thought to have, whoops, sorry about that. That was thought to have happened or lived about 2 billion years ago. And the last universal common ancestor, the cell that gave rise to all existing life forms on Earth, is remarkably old. It's like almost as old as the Earth itself, right? So the Earth is about 4.5 billion years old, and cellular life is thought to have arisen quite shortly after the Earth came to be, so 3.5 to 3.8 billion years ago. Okay, so we know that all life on Earth is related, to each other because it all looks pretty similar in the sense that all existing life forms on Earth use the same basic chemistry. They use nucleic acids, deoxyribonucleic acid, DNA, as their genetic material that carries the information from generation to generation. They transcribe DNA to make RNA, and they translate RNA to make protein. And in modern life forms, proteins are really the molecules that do a lot of the processes that we think about. They are by far the bulk of the catalysts, the enzymes that actually do chemistry inside cells. I will put up a video that you may want to watch about what is thought to be maybe the ancestral version of life, which may have originated with RNA as its primary molecule of both genetic transmission and catalysis. But now DNA has become specialized for genetic transmission, and protein has been has evolved as the dominant kind of catalyst in cells because it's very versatile relative to the chemistry that can be done with nucleic acids. And this just shows that as you hopefully remember, proteins are synthesized just like nucleic acids as chains of amino acids, but then they typically fold up into a defined three-dimensional architecture to actually do what they do chemically. Okay, so basically a cell, by definition, is a self-replicating assembly or ball of catalytic activity that can take in the energy that it needs from the environment and turn it into these molecules that encode genetic information and do work. So single-celled organisms, as I'll show you in a second, can be extremely diverse in their morphology. In multicellular organisms, there tends to be specialization where different cells take on different fates and work together to form tissues. So this is a plant tissue, a root tip. A fern is a primitive plant, but and these are different kinds of cells that have organized themselves to form a structure. This similarly is an animal structure. It's the villi that inside the intestinal lining, and these are assemblies of different kinds of cells that have arisen through differentiation and have developed the ability to work together to make more complex tissues. But even single cells can be incredibly complex. These are just some examples of just beautiful, very morphologically crazy and diverse single-celled creatures. Whoops, I showed you some single cells that were as small as a couple of microns in diameter, which is about the size of a bacterium. But single cells can be enormous. I mean, this is far from the biggest, but this is a very large, like millimeter-sized single cell organism called a stentor. This is another ciliate. These are, particularly diverse, crazy-looking unicellular organisms. And this one is particularly amazing. It has, obvious polarity. It has a foot. It's called a holdfast that allows it to attach to surfaces. It has cilia that enable it to sweep liquid across itself, and it eats, and it can regenerate if it's, here, I'll have to show you the movie again because it's pretty cool. So if you cut this in half, it will regenerate into individual cells. It's quite remarkable. So you can see inside this stentor, this is a eukaryote. It has lots of little compartments inside it that you can see in this micrograph because they are surrounded by membranes. And that is sort of the hallmark that we associate with eukaryotic cells, that they are highly compartmentalized. So this is just to kind of compare and contrast. Bacteria, which they're smaller in general than eukaryotes, although there are some extremely large bacteria, so that's not an absolute rule. But bacterial cells are generally small, on the order of a micron or two, and they don't have a lot of internal membrane structure. So They have something called a nucleoid. Their DNA doesn't occupy the full volume of the bacterium. It occupies a sub-volume called the nucleoid, which is surrounded by kind of DNA-free, cytoplasm. And they often have membranes and cell walls around them, just like eukaryotic cells. But what really is sort of the hallmark of eukaryotic cells is these membrane-bound internal compartments, and especially a membrane-bound nucleus, which contains the genetic material. And we'll talk more about the structure and function of nuclei, I think, in the next lecture. So, I mean, the term eukaryote really means true nucleus. And so having this membrane-bound nucleus is a hallmark of eukaryotic cells, although even that is not absolute, as I'll show you in a second. So this is just also comparing and contrasting plant cells versus animal cells. They have a lot in common, but they also have major differences. Plant cells are typically surrounded by cell walls made of polysaccharides, which is generally not true of animal cells. There are some differences in how they divide. Plant cells typically have chloroplasts, which makes them photosynthetic, whereas animal cells do not. And plant cells have these very large vacuoles, which play a similar role to lysosomes in mammalian cells, but the lysosomes don't tend to be as prominent as vacuoles. So let's see. Okay, so this is just another way of representing this. This is showing kind of the nucleoid, which in these images are electron micrographs. I'll have more to say about that in a minute. So they're much higher resolution. You can actually see the substructure inside individual cells. And the nucleoid in these images happens to look light compared to the surrounding cytoplasm. In eukaryotic cells, the chromatin, as we call it, often looks dark, or at least some of the chromatin looks dark in the nucleus. And these micrographs are also very good at highlighting the membranes that surround the different organelles inside eukaryotic cells. Okay, so again, like the sort of defining or eponymous feature of eukaryotic cells is having a nucleus that is surrounded by actually a double membrane. We'll talk about that, but the nucleus is one of the organelles inside the cell that has two membranes around it. That's also true for mitochondria and for chloroplasts. Okay, I wanted to, after one thing that's confusing about cell biology is, like, for every rule, there's an exception. And even though we think of bacteria as being sort of devoid of interior membrane-bound compartments, modern bacterial cell biologists have discovered quite a variety of different specialized membrane compartments. This is one studied at Berkeley by Arash Kameli's lab. These are called magnetosomes. They're little membrane-bound compartments that the bacterium makes little iron particles, which help it navigate in magnetic fields. And there's even like crazy exceptions, like this. This is a very strange bacterium that was found in Australia that actually has something that looks like a membrane-bound nucleus, and yet phylogenetically it's clearly a bacterium rather than a eukaryote. So, don't get too fixated on these like hard definitions, but in general, eukaryotes have a lot more organelles than bacteria. And a lot of what we study in cell biology is the structure and function and interaction between these different organelles. I always found these kind of hard to deal with because they all sort of look similar. They're all kind of blobby. We'll focus on kind of the function of specific organelles as we go along, and that will hopefully make them seem a little bit more different. Okay, so I said at the beginning that the whole field of cell biology has really been, has emerged from advances in microscopy, and that's because cells, most cells, as I said, are kind of on the size scale that is not visible to the naked eye. We live in this realm here, where the things that we're used to looking at are, you know, kilometers or meters or at least centimeters large, like us. And our eyes are really not very good at seeing stuff, you know, like a hair. How big is a hair? I think it's kind of in this range. So you know, as we get down to the size of cells and organelles, our eyes are not useful. And so to interrogate what happens inside cells really required the development of microscopy. So this is just the ECB version of that image. So our eyes can see, you know, a thumb, but they can't really see substructure. And modern microscopes can take us down to the subcellular level, and modern electron microscopes can take us even further to the study of protein assemblies and even individual proteins. And so I'll talk a little bit about why that is. So our eyes have a sort of finite limit of resolution, which is evolved to, you know, be useful to us in our daily life. And the resolution is limited in a sense by the structure of our eyes, by the fact that the retina, which is the part of our eye that actually records light, is quantized. It has discrete, you know, cells, photoreceptors that receive light and transmit signals to our brain to make up images. And each cell operates a little bit like a pixel on a digital camera. And so the resolution of your eye is sort of defined by the density of cells in your retina. And so for example, an eagle has a higher density of photoreceptor cells in its retina than humans do, and that enables it to see something like a mouse from, you know, half a kilometer in the air, and to pick out details in the landscape. We can't really change our eyes, but it was recognized sort of that we can see things by magnifying them, and thereby access more, you know, make more of our eye available to see the object. And this just kind of explains why that is. So if we're looking at a small thing, like a fruit fly in this case, the light beams that are kind of hitting the fruit fly and bouncing off of it, that enter our eye and go through the lens of our eye and get focused back on the retina, only those light rays only sort of hit a limited range or limited patch of cells on our retina, and that limits how much information, how much discrete information we can resolve about the structure of that fruit fly. And when we magnify an object like a fruit fly, what the magnifying does is it creates this sort of virtual image, and that actually enables more of our retina to be used to make an image of that object. And so we can make a higher resolution image of this magnified virtual fly than we can of this unmagnified fly, and so we can see more detail. And in essence, that is all that a microscope does. Although modern microscopes enable you to play with light in a variety of ways that enable you to see things that our eyes cannot naturally see by themselves. The very first microscopes that really created the whole kind of concept of cell biology were simply magnifying glasses. And this is the famous microscope created by Leeuwenhoek in the 17th century that enabled him to see single-celled microorganisms in pond water. It's really tiny. I have a little replica of this, which unfortunately is at my office, but it's really small. This is for scale. And it's just a single lens, this little magnifying lens. And this little pin is where you put the sample. You like stick something on there, and there's this little adjustment pin that allows you to move it so that the focal point of the lens is in the right place relative to your eye. And so a lot of the sort of amazing observations made by 17th and 18th century cell biologists were really through these very simple single lens microscopes. And you can see examples of them if we ever go back to campus in the lobby of LSA, or VLSB, sorry, where there's a really amazing kind of historical microscope collection. Modern microscopes are actually not really much more powerful than this one in terms of magnification, but they do a lot of things that this one doesn't do. They have many more lenses, and that enables a lot of different kinds of illumination. And also, one problem with a very simple microscope like this is, as you know, light, when it hits a glass surface, it gets bent and it diffracts, and the different wavelengths of light actually get split. And so one thing that modern microscopes do with all their lenses is they allow you to image all the different wavelengths of light and bring them to the same focal point, so that you can see multicolored images in focus. They also enable you to do, as I said, a lot of other things, which we can talk about, although it's not a main topic of this course. Of course, early cell biologists did not have recording devices like cameras or digital cameras, and so early cell biology papers were hand-drawn, sometimes using tools like a camera lucida, where the image from a microscope could be projected and then traced, or just hand-drawn based on what people saw. And so this is a timeline that's in the textbook about some of the major discoveries that led to modern cell biology, and most of those were driven by microscopy. So the discovery of these cellular structures in cork in the 17th century, and Leeuwenhoek, as I said, at roughly the same time, discovering these tiny swimming organisms in water from nearby ponds and so forth. And the whole kind of cell theory that all organisms are made of cells also came from direct observation of different tissues and different organisms. And then in the 20th century, there was a massive kind of rapid acceleration of advances in microscopy, the development of both different ways of staining cells so that they would be visible in the microscope, and different kinds of microscopes that made new things visible. And that is still happening at an ever increasing rate today, where people might have thought that microscopy actually went through a little bit of a dip in I would say the late 20th century, where people sort of thought we had gone as far as you could go. We'd hit this thing called the diffraction limit and you couldn't go much farther. And then, since then it's been realized that in fact you can go past this sort of diffraction limit. And Eric Betzig, who some of you may have met, who came to Berkeley a few years ago, is one of the people who's really kind of pushed this next generation revolution in microscopy beyond what we call the diffraction limit. I'm not going to really explain the diffraction limit in detail, but I would encourage you to look that up if you're interested. So early microscopes really couldn't, they were limited in what they could see, in that they really took advantage of just the way cells interact with light naturally. And most cells are pretty transparent. They don't have much pigment. They don't absorb much light, so that you don't see much if you look at a cell kind of the way we look at everyday objects, with like light just kind of shining on it. This is what's called the bright field image, where you simply shine a light through a specimen and look at it. And actually most cells wouldn't even be this visible, because they don't absorb light. They're made of things that are essentially transparent. But in the mid 20th century, people started realizing that if you kind of played with the light and either use polarized light or use certain kinds of tricks that would actually measure not so much the absorption of light, but the retardation of light by cells, because as light goes through different objects, like liquid, if you put a pencil in a glass of water, right, it bends the light. It's not so obvious in this case, because the air and the water have different refractive indices, and the glass. And basically the speed of light through water, through air, through glass are all different. And that's why you can see a transparent object like a glass of water, because you see basically the light sort of changing its speed at the different surfaces. And by using tricks like that, people created what's called phase contrast imaging. And this is a fancier version called differential interference contrast. So they basically designed microscopes that could magnify the small changes in the speed of light as it goes through tissue, to see these different, essentially transparent objects. And those were very powerful techniques, but they still only allowed you to see the naturally occurring kind of changes in refractive index within cells. Now, so this is another DIC, differential interference contrast image. This is still a very powerful technique, and it's particularly powerful for looking at things that are living, because it doesn't require any kind of stain, any kind of added dye or pigment. But a lot of the work that modern cell biologists do uses dyes, usually fluorescent dyes, that we can image inside cells. They can be, there are dyes that we can use in living cells, and an even broader range of dyes that we can use in dead cells, in fixed cells. And one of the kind of most powerful tools still in modern cell biology is fluorescence microscopy, because fluorescent dyes are easy to make, and by attaching them to probes, to antibodies or nucleic acid probes or different kinds of chemicals, we can basically attach those dyes to different molecules inside cells, lipids or proteins or nucleic acids, and they can be exquisitely specific and enable you to see all kinds of structures. So in this case, we're just looking at the DNA inside the nucleus labeled in one color, and the microtubules, which are part of the cytoskeleton, in a different color. And I don't know what these are. They might be mitochondria, labeled in a third color. So by kind of using combinations of tags, we can see a huge range of different subcellular structures. And fluorescence microscopy until the like early 90s was particularly useful for fixed cells. But then with the discovery and development of fluorescent proteins like GFP, the ability to target molecules inside living cells exploded. And now there's a huge range of different kinds of tools that you can use to look at molecules inside living cells, which are minimally perturbed and so forth. So these are, extremely useful tools in cell biology. This is a diagram from the book just explaining the basic kind of setup of a fluorescence microscope. If you've taken or are planning to take 133L, you will learn a lot more about fluorescence microscopy. But the basic idea is that that fluorescence is a technique where you use a dye that absorbs light of one wavelength and emits light of a longer wavelength. And so the way a fluorescence microscope works is that you use light of a particular wavelength to illuminate the object, and most of that light doesn't do anything, but the tiny amount that hits fluorescent molecules in the object, then gets emitted at a longer wavelength. And by using a special mirrors and filters, you can exclude the illumination light, which in this case is blue, and just look at the emitted light, which in this case is green, and you can see the object that emitted that green light through this process of fluorescence. That's a very superficial explanation of how fluorescence works, but the idea is that because we can target these fluorescent dyes to specific molecules inside cells, we can basically visualize whatever we want. And again, this is DNA and microtubules just stained in different colors. So as we've learned about the molecules inside cells and developed ways to specifically target them with antibodies or with other kinds of probes, we can image them. So all the microscopy I've talked about so far is light microscopy. sort of natural that because our eyes see in this range that we call the visible range of the electromagnetic spectrum, that most microscopy that we use our eyes to, that where we just magnify things and image them with our eyes is in this visible spectrum. I mentioned this concept of the diffraction limit. So it turns out that the detail that you can see in an object is directly related to the wavelength of light that you use to look at it. And it's roughly half the wavelength of the light. And so when we image things in the visible spectrum, using what we call diffraction limited microscopy, we can't separate objects that are closer together than about half the wavelength of visible light, or about 200 nanometers. And many objects inside cells, cells are often, just a few times that size. So I described cells being a few microns in size. 200 nanometers would be, a tenth of a 2 micron size cell. So if you want to see structures that are smaller than that, you have to use shorter wavelengths of light. And that's what electron microscopy does. It uses accelerated electrons, which are often down in the picometer range. Their wavelength is on the order of picometers. And so you can see objects on that size scale inside cells. The caveat, though, is that electrons don't interact with ordinary, they're not absorbed well. They're very, very high energy. They're not absorbed well by biological materials. And so in order to see things with an electron microscope, you typically have to coat them with heavy metals, which do absorb these very high energy electrons that, so when you're looking at an electron micrograph and seeing these structures, what you're seeing is a coating of metal that has been applied to typically very thin sections that have been cut. So cells are embedded in a plastic resin and then sliced very, very thin and stained with heavy metals like lead and uranium. And those are what actually interact with the electrons and create these images. But using electron microscopes, we have been able to see much smaller objects than you can see discretely with light microscopes. So again, a lot of the advances in cell biology have come through advances in both light and electron microscopy, and those are still ongoing revolutions. And this is just a diagram of how that works. I don't expect you to know how an electron microscope works. I don't even know myself, but if you're interested. And so often we use, these images are what are called transmission electron micrographs, meaning that the electrons are sent through the sample. But there's you've, I'm sure, also seen images like this, which look more like a surface scan, and those are made by a scanning electron microscope. And you can create some pretty crazy looking, this is some kind of worm with a scary looking mouth. So you can tell that that's a scanning EM because it kind of looks like the surface of something. Just at the end, I want to highlight the fact that, so in the early days of cell biology, cell biology actually sort of preceded genetics. And people just cared about finding organisms that had big cells that they could actually see and maybe look at, maybe that the organism was transparent, they could look inside it. But as we've become more interested in the molecular basis of cells and what their structures and what they do, people have moved toward model systems where we can manipulate the molecules more easily. And those are typically organisms that also have good genetic tools, because as you've learned, modern genetics, and actually going back, not even modern genetics, but going back 100 years, genetics has been a very powerful tool for making mutants and investigating the molecular basis for different processes. And so a lot of the key model organisms that are used in cell biology are also organisms that have powerful genetics and genomics tools. I think you've already learned a little bit about what makes a good model organism, but most of these are sort of obvious features, but maybe not so obvious if you haven't thought about like how to pick a model organism. But the model organisms that are most popular are ones that are easily grown in a lab, that typically require very easily made and easily reproduced growth conditions, food and light and temperature and so forth. Typically, we use model organisms that are clonal, that meaning that they've been inbred to the point that they are genetically homogeneous, so that gets rid of a big source of variation. They typically have short life cycles, that they go through cell division and even their whole reproductive cycle, often within hours or weeks rather than many years, which is like what humans do. I already said that they typically have powerful tools for genetic and genomic analysis. And often, what makes a model organism is just the fact that a whole bunch of people decided it was good to study it. And so they created a body of knowledge and resources that make it useful. And occasionally someone has decided that they want to make a new model organism, and they create a community of people, and then it becomes a model organism. So the most common model organisms that we use in cell biology, as well as in genetics and genomics, are things like, well, the mouse is obviously a mammalian model that we tend to use because mammals, we're mammals, and mice are often a good model for processes that happen in mammals that might not be accessible in other model organisms. This particular image from the book shows that this human baby actually has the same mutation in a gene called KIT that causes this pigmentation defect that you can reproduce in a mouse. Sometimes we don't want to study, like cell biology is very hard to study in a whole mouse, but we often, a lot of cell biology work is done in isolated cells, which can be grown in culture, which are, came from mammals, mice or monkeys or humans. Those are, powerful, but of course they are extracted from their natural situation. And so they may not, their cell biology may be somewhat different from the way it would function in an intact organism. And then we use a lot of other models. The zebrafish has become a really nice model for another vertebrate that's got a very short life cycle, that the embryo is transparent, so you can image lots of things inside it. Fruit flies, which are of course a very powerful genetic organism, are also really useful for cell biology. And this is my favorite organism. My lab works primarily on this nematode, C. elegans. They are transparent and have very few cells, and the cells are that they follow a very precise and completely reproducible developmental pathways. So they've become a very popular model organism. And then Arabidopsis, which is a plant with a short life cycle, is a great plant model. Yeast, both budding yeast and fission yeast, which I showed you earlier, are also really popular. They're quite small, which for me is a little bit of a hang up because I want to see more. And these cells actually, they're quite a bit smaller than the average mammalian cell. But by studying, we know, as I talked about, that all cellular life on Earth is related to all other cellular life, and so by studying processes in yeast or in flies or whatever, we can learn a lot about the fundamental processes that happen in all cells, including human cells. And we can also learn about the molecular components, although they vary a little bit over the course of evolution. Many of these molecules that execute all of the core cellular functions are shared by essentially all cellular life to varying degrees. So if you look at different proteins that do things in cells, some will be almost the same if you compare a yeast to a human. Some will show remarkable differences. So we reached 10 o'clock, and I was going to talk a little bit more about the origin of mitochondria and chloroplasts, but I will leave that for the future. And are there questions? Are there any? Yeah. Sorry if I went a little fast. I mean, it's a little hard to pace. The pacing is a little different on Zoom than it is in person. So I can't hear anybody, but I'm trying to see the chat.
There were no comments on the chat.
Okay. Okay. Looks like a few more comments on the textbook. Okay. Okay, so people are mentioning where you can get a free copy of the textbook. Like I said, I'm going to try to get the ebook made available to you guys because it's a little easier to work with than just a PDF. And I will also talk with the GSIs about using some of the smart work and other self-assessment tools that are available through the publisher.
Sorry,
Yeah.
someone asked about big quizzes about how the final will work and also big quizzes.
Okay, so I mean the final I think will work similarly to the midterm, except that I don't think I think it will happen synchronously, like everyone will probably take it at the same time unless there's a particular reason that the originally scheduled final time doesn't work for you. We haven't talked about the big quizzes, but I assume that they'll be handled sort of similarly.
Yeah, I think that the GSIs will meet to discuss the big quiz. Is my thought.
Okay, will the final still be cumulative? It will be focused on the third section of the course. I think the breakdown was something like 2/3 of the final is focused on the cell biology part of the course, and 1/3 is split between the first two parts of the course. Yes, you can ask questions during the lecture, and the GSIs will be moderating the chat, and we'll either answer the question if they can in the chat, or we'll kind of punt it to me if they don't want to do that. Yes, we will still have discussion. The mini quizzes, I think we said that we weren't going to grade those, right?
Yeah, I think it just makes it too difficult, especially if people like are in different time zones. Yeah, my understanding.
Differences be taken into account. That is a good question, since I think the final is from 7 to 10 p.m. We may be able to make it, I mean, I don't see any reason why we couldn't make it earlier. I think we want to, I think it's possible, as Nicole showed, to have a test that can be taken asynchronously and hopefully keep it fairly fair, but it would probably be easier for us if everyone took it at the same time. So we can discuss that. I guess you guys, yeah, I mean, you'll have other finals. So if we move the time, then we risk kind of moving it on top of another final that other people have, but I'll see what we can do about that. Are you supposed to do the reading quizzes on B courses? Well, I think they're largely intended as self-assessment, and so they're for your benefit. There's some additional tools that are, yeah, so you don't have to do them. They're not going to be graded, which is what Kirsten wrote. So, and right now it's kind of, let's see, will this portion of the exam be based off lecture and the textbook? So as before, like the exam will definitely emphasize what is said in lecture. I strongly encourage you to read the textbook just because, I can't say as much in an hour as the chapter can say. And I think it, because you're trying to get sort of all of cell biology now in essentially 4 weeks, I think for your own benefit, it would really behoove you to do a little bit more of a deep dive. I think, yeah.
I'm still on the thing. Sorry, I should have been on mute for a second.
Anyway, so will I be continuing the slides from here? Will you re-upload the slides for Wednesday? I think I will. I'll modify. I'll repost what the slides for today just through what I did today. I did get through. Let's see, how many did I not talk about? I'll largely pick up. I'll kind of pick up at the end. The lecture on Wednesday is focused on the nucleus, and so I think that's right. So the very end of this was really just an introduction to that anyway. So you guys should read about chloroplasts and mitochondria. Hopefully that's somewhat of a review. And we'll pick up on Wednesday with the nucleus. But I'll just post a version of the slides that is what I got to today. Any other questions? Sorry, I know this is hard. Will I be okay? I think that was the last question. But thanks you guys for being here in real time. It's a little bit disconcerting because I can't see you, and you know, but please do, you know, hopefully for some of you, especially those who are shyer, it may be easier for you to ask questions via chat than it is in the actual large lecture room. So please take advantage of that. I will be monitoring Piazza, so if you have conceptual questions, I'll be happy to answer them there, and I'm sure the GSIs will as well. And let me know if there's anything I can do to facilitate this remote learning. I know it's really hard. It's hard to stay focused. You know, I have found it very hard over the past couple of weeks to really get sort of mentally focused on anything. There's a lot of distraction. There's a lot of anxiety, and we're all kind of doing the best that we can. Yes, I will still be holding office hours through Zoom, and I'll do whatever I can to kind of make this, you know, work. And you know, okay, you're confused about the textbook. Yes, you should. Okay, so yeah, I haven't been very clear. Someone else, I'm confused whether they should be studying, taking notes on the textbook, or just skimming it as a supplemental resource. I think for the purposes of this class, you should regard it as a supplement to the lecture, but you know, this is a very abbreviated introduction to cell biology, and you know, for the purposes of the MCAT and other things, I think if you can, you know, get yourself to go through the textbook and do the questions, you'll be more prepared in the future to, you know, as a cell biologist than I can do in the limited amount of lecture and review that we have. So it's sort of up to you, but I think it will help you follow the lecture, and it will help you kind of understand the context of the limited material that I'm able to present if you also read the book. So I would encourage you to do that. Hopefully, but the exam and everything else will definitely focus on things that I have directly addressed in the lecture. Okay. Yeah, and get in touch. And I did ask people, I realized, like, if you have trouble doing the Zoom lectures, you're probably not here right now, but I did ask people, like, if you have any major, you know, barrier, if you have internet limitations or other things that make it really hard for you to access the material, let me know and I'll try to find a workaround. Anyway, I will see you if you come to office hours, and I don't know, I guess there's not going to likely be much discussion this week, but I will hold office hours anyway, and I'll see you at lecture on Wednesday, and I will post the recording of this one to B courses. All right, take care, everyone.
Thanks, Abby.